Hi, it's Dr. Robert Psychiatry. Welcome to a new episode of Doctor Podcast. Today, I'm pleased to have an excellent guest, Dr. Herb Lachman. Dr. Lachman is a professor of medicine and psychiatry at the Albert Einstein College of Medicine here in New York City. And you can see his information here behind us. Dr. Lachman is also an associate professor of neuroscience and genetics, also at the Albert Einstein College of Medicine. Today, we're going to discuss some exciting topics about genetics and psychiatric and mental disorders with Dr. Lachman. Herb, thanks for coming today. Really
Great to see you
appreciate it.
again.
Yeah, it's a horrible rainstorm out there. I got drenched. My suit is soaking wet, so thanks.
I came early so that I would avoid. I came really early. I'm one of those types who comes to airports really early.
Well, that's good. Hopefully the rain clears up. Now, Herb, you have a fascinating background in that you trained in internal medicine and then did a fellowship in genetics. I'm sorry, fellowship in hematology.
Right.
And oncology,
Right.
but somewhere along your career, you switched to neuroscience and psychiatry and genetics. Can you tell us why?
I made a bad mistake in my life. So I was a cancer researcher early on, and I was studying two cancer genes, c-MYC, MYC, and P53, and it was very exciting. A great time to be a cancer biologist because all these oncogenes were being discovered. And then I realized that I was kind of a small fry. I was just starting out. There were really great investigators, Nobel Prize winners in this field, and I began to get a little antsy that maybe I would not make a major contribution. And around the same time, a couple of papers came out in Nature and Science, pointing to regions on chromosomes 5, 11, and X as having genes involved in schizophrenia and bipolar disorder. Those were false positive findings, it turned out. But it was enough to get me really excited about thinking about these genes that can cause these incredible changes in a person's behavior. So that sort of got me started to think about bipolar, especially. I became a little bit obsessed with it, and I just tried to play around with models that might result in these drastic mood changes. And eventually, I was able to convince the chair of psychiatry at the time, Dr. Herman von Prague.
That's at Albert Einstein.
Yeah, a really great early biological psychiatrist to start a psychiatric genetics unit. And he appointed me as head, along with former classmate Dimitri Popoulos. And that's how I got started. And at the same time, Dimitri was involved in this disease called VCFS, Velocardiofacial syndrome. It's got 5 different names. It was discovered at Montefiore. And it causes a whole slew of congenital anomalies and cardiac defects. And about 1/3 of those patients have a psychiatric disorder, schizophrenia or bipolar. And it's due to the loss of 50 to 60 genes on chromosome 22. And one of those genes is a gene called COMT, which I began to study. And I created kind of a cottage industry of COMT-related disorders that still persists.
So you've discovered genes associated with bipolar disorder. What about schizophrenia and autism? I know you're interested in that as well.
The COMT gene ended up being a variant. I discovered a common variant in that gene that causes a major change in activity, and people began to look at that in schizophrenia, bipolar, Parkinson's disease, everything, psychopathy, pain, sensing pain, and it turned out that it only has a few effects on people. And one of the problems is that in order to prove that a genetic variant is causing a problem, you need to have very large sample sizes, and we didn't really appreciate that until 5 or 6 years later. Anyway, I ended up not. I ended up doing work on disease modeling with these things called induced pluripotent stem cells. That's what I've been doing for the last 12 years or so.
Right. Stem cells are a hot topic
Yeah,
these
that's right.
days.
Yeah.
Back in the old days, from what I recall in my training, most psychiatric illnesses were thought to be sometimes familial but more environmentally induced. Is there increasing evidence that there's a genetic basis for them and certain environments trigger the clinical disorder?
I tell psych medical students who want to go into psych, that if they thought they would escape genetics, they were wrong. It turns out that autism, schizophrenia, and bipolar have a very, very strongly genetic. Maybe 80% of the variance is due to genetics. Genes really are the main cause of those illnesses. There are also maybe environmental factors that have an impact, but it's primarily genetic. And now we know. Now we have identified, or my colleagues have identified, hundreds of genes that cause each of those. It's a very, very complex trait.
So it's not just one gene, it's a
Right.
combination of various genetic defects that in combination cause the clinical disorder?
Right. So in any one person, you might have two or three genetic variants that are doing it, and then someone else, they might have genes A, B, and C, and someone else might have genes, C, D, and F, and so it's not everybody doesn't have these hundreds of mutations. They have only a small handful.
A few of them.
Yeah.
Yeah, there are some eye diseases as well, similar to that. I'm an ophthalmologist,
Yeah.
as you know, where, for example, retinitis pigmentosa has a variety of different genes that can trigger it. Are there tests available for these genes? Can someone have a genetics test to see if they have the schizophrenia or autism or bipolar gene?
Well, in autism, the standard care is to do a genetic analysis, typically for Fragile X, which is the most common cause of autism. Maybe 5% of all autistic cases.
That's a gene defect,
Yeah,
the Fragile
Fragile
X?
X, yeah.
Mm-hmm.
And so kids are tested for that, and then they are analyzed using these things called arrays, CGH arrays, which look for copy variants, which are these large chromosomal changes. And then after that, they can get DNA sequencing, exome sequencing, which is DNA analysis looking for specific mutations in the coding regions of genes. That's standard care. And it's not that we can do anything about it now, except we can tell the families. They're relieved. They want to know what's going on.
Right.
They can use that information in some cases for family planning. But it hasn't trickled down as much in the adult psychiatry community. They don't. So people are not really getting analyzed that much. Some are. It's changing. But I think it's very useful to get these studies done because families now know it wasn't a drink that I had when I was pregnant. It wasn't this infection. I have this gene, or in some cases, it's a de novo mutation, meaning that it happened in the germline, and now we know. So families can form groups, Facebook groups, and start nonprofits, and it's very, very important to have that information.
Speaking of groups and nonprofits, you do a lot of research on these illnesses, and people can contact you, right, with this information in the background. So
they can email me, and I'll be happy to forward them more information.
Right. So if anyone in the audience watching this has any family members who has any of these psychiatric illnesses and is interested in more information, or possibly anyone who's interested in providing funds for additional research that Dr. Lachman is doing, please feel free to contact him with the information in the back. You mentioned autism and genetics. I've asked a few guests already about autism and relation to vaccination. They all told me that there's no connection to vaccines. Do you agree with that?
Oh, totally. I mean,
Okay.
the Wakefield study that started this whole thing was an unbelievable. It was a mistake. I don't know how it got past the reviewers, how it got past the editors. It was a horrible paper, and scientific fraud was committed. Ethical violations occurred. It was really an amazingly poor study, and it was retracted by Lancet. And then many studies since then showing that there's no connection at all. The problem is everybody gets vaccinated as a kid, and a lot of kids, autism is very common now, 1 in 40 or so. And every now and then, a kid will have a regression, and it's temporarily related to them getting a vaccine. It's like us, when you have elderly parents, you always dream about your parent dying. Every now and then, a person in the world is going to have a parent die on that same day, and you'll think, oh my God, I dreamt that my parent. But no, it's totally coincidental.
So it's a
Yeah,
coincidence. There's no cause and
absolutely.
effect.
Yeah, and not only that, these vaccines actually improve the brains of kids. So back in the day, before the MMR vaccine was used, that's mumps, measles, mumps, and rubella. There were 3 million cases of measles every year, and 1 out of 1,000, or two out of 1,000, died or developed brain inflammation called encephalitis, and that leads to brain disorder,
Right.
autism, and other cognitive abnormalities. And also back in the day, we had rubella, German measles, which is a fairly benign disease. But if a woman gets it in the first trimester of pregnancy, the child would have congenital rubella syndrome, which also causes uh, brain damage.
Right.
And I just estimated, since MMR
Mm.
vaccines came into use, maybe 40, 50 years ago, I think that maybe 500,000 people in the United States alive today have normal brains because their parents and they were vaccinated.
Well, that's good to know because unfortunately there's still bad information out there in the media about the MMR
I
vaccines.
have an
And
anecdote, if you don't mind, about
sure.
the. So I was dealing with a family with bipolar disorder, and doing a genetic analysis, and the son of one of the individuals was at the house. I was drawing blood for DNA, and he didn't have bipolar, but he was diagnosed with Asperger's, which is now part of the autism spectrum. And he was a very nice young man, but I kind of suspected something was up. So I did a chromosome analysis on him, and he had a duplication of a region on chromosome 8 involving some 200 genes. Very rare, but well known to cause a some developmental issues and Asperger's. And he was having trouble with math. And he was getting tutors, and the parents were really frustrated that he couldn't get past a certain level. So I called the parents and said, "Look, it's good that you're getting this tutoring, but there's a window here because your son has this chromosomal abnormality that's well known to actually cause math problems." And over the phone, one of the parents said to me that I was wrong. The reason why he had this problem was that he was vaccinated as a kid. So
Mm.
even when you're given the genetic information,
Right.
so strong is this belief about the vaccines that it just makes you delusional about it. So,
The media has tremendous influence on what people think and what they know or what they think they know.
well,
So,
these they have these narratives. These narratives are powerful,
right.
and they're more powerful than stats.
Right. More powerful than
Yeah,
science. Another thing I want to discuss with you is you mentioned stem cells earlier. I had a guest a few weeks ago, Dr. James Griffo, who's the head of the fertility center at NYU Langone Medical Center here in New York. He mentioned that he is creating 5-day-old embryos, which are stem cells, and then implanting them in the uterus of
right.
women who
Hmm.
can't get pregnant naturally. So you're doing stem cell
Right.
research. You mentioned they're induced pluripotent stem
Right.
cells, which I'll just call IP stem cells
Uh,
for short. These are similar, but not the same as embryos. Can you explain why you're doing that and what the issue was with using stem
yeah,
cells
sure.
from embryos?
So the structure that's implanted in women who need to conceive with in vitro fertilization is a blastocyst, which is about 200 cells, a 5-day, 6-day-old embryo, and ththose contain ES cells, embryonic stem cells. Those stem cells have this incredible capacity to differentiate or turn into any cell type in the body. So ES cells become heart muscle and brain and neurons and bone and so on. So they have this intrinsic capacity. And of course, you grow into a human being from that. And in 1981, uh, researchers were able to isolate the embryonic stem cells from mice. And those were used to create these mouse models of disease, which we now have thousands of them. And it led to a Nobel Prize in 2007 to the researchers. In 1995 or so, researchers at the University of Wisconsin-Madison this isolated human embryonic stem cells. And these were touted as being useful tools uh, to replace dead or damaged tissue or cells in people. So if you have a spinal cord injury, or if you have ALS or Parkinson's disease, these cells can turn into the appropriate cell types that can theoretically at least replace the damaged tissues.
So you could make nerve cells
You
or
can make,
kidney cells, heart
any
cells,
cell you
whatever
want,
you
any,
cell
yeah,
you
any
want.
cell you want. And the problem is that when you do that, you are these embryos, by the way, are obtained with informed consent by parents who don't use them anymore. They get the one or two implanted, and the rest are not used. So they're frozen in these facilities, and they donated these samples. But among the ultra-right religious community, these embryos, these 5-day-old balls of cells, are human embryos, and if you destroy them, which you need to do to make human embryonic stem cells, you're killing a person. So that has created this climate in the U.S., making it very hard to do that kind of research. You can still do it, but there were times when there were federal bans on making new stem cells. Some states have bans now. So if you're doing work in this area of research, you don't want that undermined by politics.
Right.
So we needed to come up, the field needed to come up with a new technique to produce cells that mimic ESLs, but would not involve the use of an embryo.
Right.
And that's where these IP stem cells were discovered. And they were discovered by Shinya Yamanaka, a Japanese researcher, who provided the technique for making them from skin cells. You can take skin fibroblasts and turn them into these stem cells that resemble embryonic stem cells. And those are the cells that are now used primarily are being investigated to use for replacement therapy.
So there's no controversy because there's no embryos involved.
Right. And they're better in some way. Well, in many ways, they're better because they're derived from the person.
Right.
And if you use embryonic stem cells, you might have, and you create, let's say, dopamine neurons for Parkinson's disease, you would need to treat that person with immunosuppressants. And if you're using your own cells, then that's,
Right. The body doesn't reject
Right.
their own cells, even if you grow them in a test tube
Yeah.
or an incubator of some
But it's
type.
really expensive to do that.
It's very expensive.
Yeah, and it's
Right
actually,
now, in a few
no,
years.
well, I mean, you have to grow these cells. It takes many, many months. If you're growing them from patients, it costs hundreds of thousands of dollars. And for disease like Parkinson's, which is a public health disease, and 1 million people have it,
Right.
1 million times 1 million, it's a lot of money. So it's not really feasible in the long run. But
So
it is
only the billionaires can afford it right now.
the way it is now. Yes, right.
I see. How do you direct a IP stem cell, not an embryonic stem cell, to become a certain organ or tissue? Is it a chemical? Is it some sort of other magic potion that you use to tell that stem cell to become skin or bone
There's
or
a
kidney?
little magic to it.
A little magic?
Yeah, but so the people who do this, and I'm basically following other people's recipes, so as I don't do this work.
Now you make neuronal stem, right?
we make
Which is
neurons,
a type of a brain cell
Right.
from a stem cell.
That's right. So basically, what is done is you try and mimic what's happening during embryogenesis, and you add growth factors to induce differentiation. You add certain inhibitors of pathways that would keep the cells as a stem cell, and you play around with the recipe until you come up with one that produces a very nice yield of the cells that you want.
I see. So you try various chemicals and see what they do and how that cell is directed, and then you
Right.
keep on improving on
Well, the
that.
people who made those first discoveries did that. I just followed the recipe.
I see.
It works. So that my job is not to do what they did. My
Right.
job is just to copy the recipe and then do my research from that.
So you don't reinvent the wheel. You
no,
know how
I
to
would like to. I wouldn't mind doing that. But that's I don't have a lab big enough of funding to play around, to tinker with.
Well, after this program, when people contact you with this information and want to donate millions of dollars to your labs,
that would be nice, right?
perhaps you can move along. So is it possible, let's say you had enough money, or let's say 5 or 10 years from now, you could just grow brain cells Or if somebody needs a kidney, could you actually grow that in the lab and then replace that as an organ?
So the work that we do is we study these neurons that we make from IP stem cells from patients. So we grow patient specific neurons.
Where do you get those cells?
We
From
get
what part of the patient?
I get them from blood.
From
So
blood.
so we take white blood cells, or skin sometimes, and we turn them into stem cells using the Yamanaka formula or current formulas, and we grow these cells, and then we can differentiate them into neurons and other cell types, and we study. So we're not growing these cells to replace anything.
Right.
Other researchers are doing that. We are just creating models of disease. So we're studying what is the effect of these schizophrenia and autism genes on the cell, on the neuron. How is it affecting other proteins and other genes? How is it affecting the electrophysiological properties of these cells? And the point there is to find these pathways that you can then, where you, after which you can find drugs that might block or reverse the abnormal pathway.
So you could take some skin cells, for example, from somebody who has schizophrenia and a genetic defect that
Right.
you've identified. You can then grow those cells, identify where possibly the error is, and maybe that will help come up with ways to treat that in the future.
Right. That is what we do. That is what many labs do. And there actually are some phase 1 clinical trials being carried out now to test different drugs that were found in various diseases.
So it allows you to target treatment very specifically for individuals as opposed to now where people are just given medications that hopefully work for them that are not specific.
Right. So this is precision medicine. The idea,
Right.
yeah. And this is and in these disorders, we have hundreds of genes. It's very hard to find one treatment that'll fit everybody. So that kind of medicine, that kind of treatment, dealing with the person's specific genetic abnormality, is going to be really critical.
How long do you think it'll take? Next 5, 10 years, we'll see very targeted treatments for specific disorders?
I tell you, I'm really bad at making those. I've been bad. I've been off so much on predicting things like that that I try to stay away. But now that we're doing
How
the
are you in the
podcast,
stock market?
my wife handles that.
All right.
So I mean, the technology is so amazing now
Right.
that I think that within a few years we'll have some therapies. The problem is dealing with a disease that occurred during brain development, and how much of that is cemented into the brain's anatomy. How much can you reverse?
Right.
And this is the big unknown. But we know from work done by non-medical people who treat kids with autism and other developmental problems, who get the one-on-one occupational therapy and speech therapy and this and that. Those workers have done something incredible. They've turned these kids, that they've maximized what they can do. They've turned kids who would end up being institutionalized into kids who can live on their own in some cases and work. I mean, Down syndrome, there are lots of kids with Down syndrome who are highly functional, and that's
Right.
only because of these non-medical people. And that means that the brain is plastic enough so that a person like me and my colleagues can find a drug that might mimic or enhance what these one-on-one therapists do. So I'm optimistic that we can do something. We're not going to turn them into Albert Einstein necessarily,
Right.
but let me tell you something. If you can get somebody with Down syndrome to travel independently and to live with a roommate,
Huge accomplishment.
This is gigantic.
Right.
Gigantic. So that's the goal.
Right. I just read a few days ago that some researchers actually got human kidney to grow inside of a pig by modifying the genes. And the hope is that in the future we'll be able to grow full-fledged kidneys or other organs in pigs which have a similar anatomy to
Right,
humans. and then take that organ from the pig and then put it into the human.
Well, another possibility is to create pure human kidneys in the test tube. So there are
Right.
kidney organoids that are being made. They're very small. They can't do what a kidney does, but they do resemble uh, kidneys. They have partially functioning nephrons, and at some point, maybe growing those cells on some kind of scaffold might produce a functional kidney that can then be implanted. And again, you would be using the patient's cells, so
Right.
you wouldn't get rejection. That I think is not so. That's not science fiction anymore. That's doable.
Yeah, at NYU, I think they transplanted a modified pig kidney into a human.
Yeah,
This
they
is
humanize the pig kidney. That's one approach. You
right,
still have a rejection situation, but I mean,
right.
anything you can do that will give you, organ transplant. There's a shortage of donors. So, yeah.
Right. So if you can use the patient's cells, grow the organ that you need,
Yeah.
and transplant it, you avoid the entire rejection problem. You avoid all the toxic anti-rejection
Right. No, it's
drugs.
something that I think will be done. I all this stuff. It's like the discovery of the invention of the airplane. I mean, you had the Wright brothers, biplane.
Right.
Now we have these jets. It
Right.
took a century.
Right.
We're now at the Wright brothers stage of development in many of these treatments.
That's a great
Yeah,
analogy. But things are moving
things move
faster.
much faster. Yeah,
Right.
right,
So it
right.
could what took 100 years may take five
Again, no predictions for me, but
right.
yeah, it's coming.
Which leads me to the next discussion of gene editing using CRISPR technology, which is in the media almost every
Right,
day.
right.
I read about it. Can you tell us about CRISPR technology, what that does, and what the future of that is, and how you're using it?
Yeah. So CRISPR is, fundamentally a gene editing tool, and basically using CRISPR, you can you can knock out a gene, or introduce a patient mutation in that gene, or correct a patient mutation to the normal variant. And this is all due to the ability of CRISPR to target very specific regions of the genome. And it's the reason why it took off is that the other techniques used to modify the genome in the same manner were really, really complicated. Really, I mean, it took months to create to do that. CRISPR is done really easily. It's a one-step process, and it was discovered by many investigators, but Jennifer Doudna and Emmanuel Charpentier were the main discoverers. They won the 2020 Nobel Prize in Chemistry for that, and the technique is so elegant and so simple. And the thing that's amazing about CRISPR is that it is derived from microbes. It's a bacterial immune system, and maybe one third to one half of all bacteria have evolved a CRISPR system. So we've taken this, and this system is meant to kill incoming viruses. Even bacteria are infected by viruses, and we've taken this primitive immune system. Well, not so primitive. It's a pretty advanced immune system. We've taken this system in bacteria, big bacteria, and now we're editing genes and curing sickle cell disease with CRISPR, which has
Right.
been done in a couple of patients. So this is really inventive use, evolution of a basic science discovery into clinical application. This is the power of great science.
So basically, you're able to take out pieces of DNA from a gene, or put
Put it
in
in, right,
new
or patch it. Yeah,
pieces,
right, exactly.
or put them wherever you want in combination. So once we figure out the entire genome, which I think has pretty
We've
much
done
been
that. Right,
figured
right, right.
out, you can, once you identify what every piece of DNA does, you could possibly edit it, change it. So if somebody has a disease because they have 20 abnormal DNA segments in a gene, you could potentially take those out and put in the correct gene.
Right. So yeah, it's being done. It's being done now.
It's like Microsoft Word
It is
for
you
genes.
delete, insert. Yeah, it's
Right.
the same idea. So
Copy
so
paste.
so gene therapy is done on somatic cells. Those are cells in the body.
Right.
They don't get into the germline. They don't get into sperm or egg. So we can take cells like stem cells from the blood, and we can correct the sickle cell mutation, give it back to the patient, and they don't have sickle cell disease anymore. And you can do that now for many, many disorders of many blood cell disorders. It's much harder to do in other cell types, but even then, it's being done with these viruses that are engineered to target uh, specific organs and cell types like the liver or even the brain. But it's very easy to do it on stem cells. You just isolate them from the person, do the CRISPR correction literally in a test tube, and then inject it back into the person. You can't do that for a liver disease or brain disease. You have to
Right.
have another delivery system. But that's being done.
What if you took an embryo at, let's say, the 3 or 5 day stage and you knew there was a genetic defect there? Could you potentially take one of the embryonic cells, modify the DNA, get rid of that defect?
Well, you can do it in theory, but in practice, it's not done because there's a ban on doing germline intervention.
And why is that?
Well, one reason is that CRISPR has problems. One of them is so-called off-target effects. So you can direct the CRISPR complex to a specific site in the genome, but it also is so powerful it can knock out other genes. So that's one problem that has
Right.
to be resolved.
So we don't know all the gene to gene interactions. In other words, if you change gene A, what effect might that have on gene B and C
Yeah,
and D
right.
and
So
E?
that's that, and there's also a reluctance to manipulate the human germline. That's the main obstacle right now.
Right, because
And
then people want to have 7 foot tall kids who play for the
right,
NBA,
right,
right?
right. We can do that for the Knicks. That would be actually, I'd be in favor of that happening. Well, now, or maybe getting a big lineman for an offensive lineman for the Giants that we can engineer a few of those.
Right.
That would be very
So if you
helpful.
want a 7 foot child who plays for the WNBA or NBA, contact Dr. Lachman and
Yeah. So
he'll
there's a
arrange that.
reluctance right now to do that, and there has been a case where that was done in China
Right.
a few years ago, where this investigator um, made manipulated fertilized egg in twins and knocked out a gene called CCR5. CCR5 codes for a protein that's the receptor for HIV.
The virus, HIV
Yes,
virus.
so HIV gets into the cells by binding to CCR5. And we know from genetic studies that there is a variant found primarily in Europeans where CCR5 doesn't work and doesn't form. And HIV does not get into those cells. You have two copies of that. And that's found in about 1% of Europeans. You are resistant to HIV.
So those people are immune to getting
They
HIV.
can get it, but it doesn't really grow. There are HIV strains that use another receptor, so it
Right.
won't target those. So there's a very good evidence that can be done. The problem is that it was medically unnecessary because the father was HIV positive, and there are techniques to reduce the risk of infection if fathers are infected. There's absolutely no reason to do that.
I see. He
And
was put in jail for a
he
few
was
years.
jailed for 3 years
Right.
and fined, and no, he wasn't the first person in history whose hubris got in the way of common sense, and it led to really nasty consequences.
Right.
So right now we're not doing germline manipulation.
Right. Good idea. I've read some controversial information that maybe CRISPR was used to modify or create the COVID virus,
Oh boy,
possibly. Is that
no, definitely not.
definitely not?
No. The COVID virus, SARS-CoV-2, is too clever to be engineered by humans.
Really?
Yeah, we have never. We can actually create a whole organism from scratch. We
Right.
can do that, and a whole microbe, bacterium. But you can't create a new one. No one knows how to do that. Nobody. And SARS-CoV-2 has features that nobody could have invented. So SARS-CoV-2 is a natural virus.
Just a mutation that
Yeah,
occurred, and
like
then
like all of the mutations that they occur in nature, and they mutate quite rapidly, and every now and then you get a mutation that likes human cells, and that's what happened here. Now there may be other issues with SARS-CoV-2 that we don't need to get into, but
right.
it was not engineered. That I can guarantee that.
All right, you heard it from Dr. Lachman.
Right.
Another topic that I want to discuss that that I know you're doing research in is basically psychiatric illness that occurs secondary to a strep infection, like strep throat. Now, strep throat is one of the most common infections that kids get and that pediatricians see, and typically it's treated and it goes away. Rarely it can cause heart or kidney problems if it's not treated promptly. But now in the last few years, there's a new condition that's been discovered. Tell us about
Yeah.
that.
So the acronym is PANS, Pediatric Acute Neuropsychiatric Syndrome, and the strep variety is called Pandus. That's what was the first entity that was discovered maybe 20 years ago. And it was found that kids were developing acute onset OCD, obsessive compulsive disorder, following strep infections, and they responded to antibiotics.
How soon after the infection did they get
quickly? Quickly. It's sometimes overnight, usually within a few days.
Wow.
And sometimes the parents describe a light switch going on. And after a few years, it was realized that strep was not the only microbe that was inducing these acute syndromes, and the Pandus was PANS diagnosis was used to as an umbrella term for strep-induced OCD and other infectious disease-induced psychiatric symptoms. And it's very controversial because a lot of doctors don't believe in it. There are no inflammatory markers. If you do a scan of the brain, an MRI scan of the brain, you don't see inflammation. And it's supposed to be a neuroinflammatory disease. But it turns out that MRI scans are very often negative, even with neuroinflammation. And there's a lot of pan skepticism. And it's not easy to distinguish between OCD occurring after infection with OCD occurring in kids as part of a psychiatric illness. But it turns out that PANS is not just OCD. It's a combination of OCD, restricted eating, anxiety, rage, autonomic nervous system problems. 1/3 have joint inflammation. There's a whole slew of neurological and psychiatric problems. Very different from.
What's the evidence that it's related to a strep infection? Is there blood tests or serology that shows that?
Well, for the ones that are strep-induced, they usually have a rising titer of strep antibodies.
So if you do a blood test, you can see antibodies.
But kids have strep.
Right.
It's not that easy. It's really not that easy. Right. That's one of the reasons why it's controversial.
I see.
Do
you believe it's a true entity?
Oh, there's no question about it. I mean, we actually found some genes that So no, I'm heavily involved in this condition now. So my group and my co-investigators and many other groups are now doing gene sequencing to look for underlying mutations. And we published a paper a year ago where we found 11 genes that with very, very rare mutations that cause PANS in 21 individuals.
So are you saying that certain individuals with a certain type of gene are just susceptible to this effect from a strep infection? If they have the right kind of gene, get the right kind of strep, they get this?
Right, and it's not just strep, it's many infections. Other viral infections, mycoplasma infections. So you have, some of the genes are regulators of the immune system. So the idea here is that you have an infection that's triggering an overactive immune response. Some of the genes code for proteins that are seen in autism, and it's triggering inflammation, we think, through another mechanism. And it turns out that a lot of kids with autism have superimposed PANS. Mm. And you can treat the PANS aspect. You won't treat the autism, but you can treat the acute onset problems with either antibiotics or steroids or intravenous immunoglobulin.
Really?
But the response is
antibiotics and steroids.
Yeah, but not everybody responds to that
Right.
because it's
So,
genetically very heterogeneous.
right.
Going back to the precision medicine idea,
Right.
and
If
that's
there are
what.
people in the audience who have family members with PANS, can they
Oh,
contact
they
you
should contact me. I'm the go-to person
for
for that,
this
and yeah.
research?
So
Can you then take skin cells from them, stem cells, and then investigate them and see what genetic defects are present?
well, we're doing that now for one
You
of the
are.
variants. So we've actually made stem cells, and we are growing microglia, which are the brain's immune cells, which we think is the cell type that's involved in transmitting the infection and the immune response in the periphery to a neuroinflammatory process. So we're doing studies on that. And I'm trying to get funding now from the NIH to look at another gene that. So we're trying to. It's very tedious work. It's very complicated work.
Right. The, tell us a little about the funding process. How easy is it to get money from NIH and other government sources as opposed to private sources of funding?
You know, people have a misconception about what scientists do. They look at people like me and maybe, we have lab coats and we're pouring contents of one into another and we have smoke all over the place.
Right.
People who run labs like me, most of us don't do research. We have postdocs and technicians and grad students who do that.
So you have assistants and
Right,
colleagues,
right.
right?
We are fundraisers. We
Really?
spend our time writing grants, rewriting grants, reviewing other people's grants, writing papers, reviewing papers, getting back comments from our papers, and revising those papers. We are at the computer doing this, and
Right.
a big part of the job is getting money, and the NIH funding stream is very, very tight. Real money the NIH budget hasn't really changed that much. It goes up a little bit, but it doesn't keep up with inflation. And right now, if you're writing a new grant, the chances of getting that grant are 10%.
Really?
Right.
And it's a lot of work to do these grants.
It's your whole life.
Really?
It's unbelievable. And then when you get back comments, the comments sometimes are very helpful. Sometimes the comments are absurd, and you can't respond to them like you would respond in a review and say X, Y, and Z. This is not correct. You have to wait for the next cycle, next grant cycle. So you write a grant, you get a rejection, and then it's a year later that you're writing second.
It's interesting. I've submitted occasional papers to medical journals, and occasionally the reviewers give me these absurd comments that are almost impossible to discuss or debate. So it sounds like you have a similar problem with funding.
But at least you can revise the paper,
Right.
and it goes right back in. Not for the NIH. You have to address the comments, but wait for the next cycle to
Really?
yeah. So we have some funding from the NIH, and we have some funding from private donors interested in PANS, and I work on a disease called Low syndrome, which is very rare, and I gotten funding from the Low Syndrome Association to do this research. You can't. It's very hard to get money for rare diseases.
Well, if there are any people in the audience with any of these conditions that are interested in helping fund Dr. Lachman, please contact them. Now, I understand that in addition to all this research and writing grants and typing away, you also do a lot of teaching at the Albert Einstein College of Medicine.
I do a lot of teaching. Yeah. So I teach human genetics to the med students. We just finished our session, incoming students, graduate students, public health students, neuroscience students, and psychiatry residents. So I have a very active teaching load. So but I enjoy that. So that's okay. And then we have administrative things to deal with. I see patients a few days a month. So I'm really at my desk from 6 to 7 a.m. until 7 or 8 p.m. with a break, meals, my wife, and working out. It's a full time. It's really a full time job. Sunday through Monday. It's
7 days.
It really
a week.
is. Yeah.
All
But it's
right,
not
well,
work because it's great stuff.
yeah, if you're enjoying it, it's kind of like a hobby.
To be a scientist now is just incredible. There's so many tools. You can do so much. It's really incredibly exciting. I envy the students who are coming in now
Are students now as motivated as we were years ago? Do they have different interests? What's your feeling
Well,
on that?
well, right now there are very few medical students who are doing what I did, which is to train in medicine, do a postdoctoral fellowship, or get a PhD, uh, and go into research. People who go into research are MD, PhD students.
I see.
Strict MD students are not going into research, and a major reason for that is the cost of medical education, which is astronomical and much higher
Right.
than when we went to school. So they want to just get through it and finish their residencies and make money and not train anymore. So there are very few MDs, strict MDs, who are doing basic research.
So they go into clinical medicine.
They do clinical work or
take
clinical
care.
research,
Right.
Or they get MD, PhDs, which really weren't available in our day,
Right.
and they take that route. And even those students, many of them go into medicine. And the students, I mean, like they're idealistic. They are energetic, especially the first month. I mean, it's just it's really great to see a full class.
Right.
By the end of a few months, you see nobody's there. They just watch slideshows at the leisure. But at the beginning, they're all attentive. And,
They're all there.
yeah, so it's fun to be with them.
Great. Well, this has been a very fascinating discussion. We learned a lot about genetics, psychiatric disorders associated with genetics, and some new
Stem
syndromes.
cells, CRISPR, we covered everything.
We pretty much covered everything.
The Knicks and the Giants and
Right.
literally everything. So.
All right. Well, thanks very much for coming today. I really appreciate it.
It's great to see you.
All right. Great to see you too.
Yeah,
Take care.
Very good.